Our mission here is to slice through complexity, find the most crucial, fascinating details, and deliver the knowledge you need to be genuinely well-informed.
And today, we are wrestling with a profound biological paradox, really, and it's all embodied by a single creature.
The sea lamprey, Petromyzon marinus, it's an animal that, it forces you to try and reconcile two completely opposite ideas at the same time: eradication and conservation.
That's it exactly. I mean, if you're anywhere near the Laurentian Great Lakes of North America, this animal is public enemy number one.
It's the vampire fish, right? That's the name it earned.
It did. And for good reason. It's a catastrophic invasive species. Its suppression is managed by this multi-decade, highly funded international control program.
And it sustains this massive economy we're talking about, over $7 billion a year.
Right. But then you flip the map.
And it's a completely different story. You cross the Atlantic to its native range, the Eastern Atlantic, the Mediterranean, and this exact same animal.
Yeah.
It's not a scourge. It's an imperiled species.
Threatened or endangered in many regions. It's facing huge environmental pressures, habitat loss, all the things we associate with a species we need to protect.
And historically, it was a delicacy. People ate this.
Oh, absolutely. A sought-after cultural delicacy. Pickled in Finland, served as a high-end seasonal dish in Spain and France. It's just a completely different reality.
So this deep dive is really about understanding that duality.
Yeah.
How could a single ancient species become this ecological weapon in one place?
While needing to be saved in another, yeah. Our goal here is to uncover these specific, almost miraculous ecophysiological adaptations that allowed it to just devastate the Great Lakes ecosystem.
And the flip side, how modern science is exploiting those exact same features, its physiological vulnerabilities, to keep its population suppressed.
We're grounding this deep dive in some really detailed scientific sources, primarily from a synthesis on sea lamprey ecophysiology published in Frontiers in Fish Science, but also public domain resources from the Great Lakes Fishery Commission and other top academic reviews.
We're basically connecting its 450 million year history to its very, very complicated life today. So let's start with that history. And when we say history, we mean deep history.
Deep, deep history.
When we talk about the sea lamprey, we're talking about a vertebrate that predates, well, it predates dinosaurs, it predates land animals, it fundamentally predates jaws. This is a true living fossil.
That's exactly right. Lampreys and hagfish are the only two living groups of jawless fish left. They're formerly known as agnathans or cyclostomes, which just means round mounts.
And the lineage is just ancient.
Yeah, incredibly ancient. They diverged from the main vertebrate path roughly 450 million years ago.
Oh.
What's interesting, though, is that while some of the earliest lamprey fossils are from, say, 360 million years ago, the predatory parasitic lifestyle we see today, the one with the suction disc that latches onto hosts, that's thought to be a slightly more recent innovation.
More recent being, what, 100 million years ago?
Yeah, something like that. Maybe mid to late Jurassic period, 150, 160 million years ago. So the vampire behavior is ancient, but the jawless body plan is even older.
That context is just so crucial because despite that immense age, all modern lampreys follow this remarkably consistent and vulnerable life cycle.
And that cycle is everything. It's what defines its success as an invader, and it's also what makes it a target for control. There are 4 main stages.
Okay, walk us through them.
The first stage, which is the longest and for them the most vulnerable phase, is the larval phase. At this point, they're called amocoets.
And this isn't a quick phase. This is a marathon, not a sprint.
Absolutely. Depending on the stream temperature, the productivity of the river they're in, this phase can last anywhere from 3 to 7 years, sometimes even longer.
And what are they doing for all those years?
They are burrow-dwelling suspension feeders. They just hide away in soft, silty substrate, drawing water across their gills with a muscular pump. They're eating detritus, algae, bacteria.
So mud. They're eating mud.
Pretty much. Over 90% of their diet is detritus, just the organic muck on the river floor. So for most of their life, they're actually benign mud dwellers.
And they're blind at this point, too.
Largely blind, yeah. They have very poorly developed eyes during this stage. They're relying almost entirely on chemosensory cues, their sense of smell, to orient themselves.
And this sedentary lifestyle, this hiding in the mud, that's the key for the control program later on, isn't it?
It's the absolute key. It concentrates them in predictable locations. They're concentrated and vulnerable, totally unlike the highly mobile adults.
Then comes the change, the transformation that turns this blind, filter-feeding worm into a high-powered predator. Metamorphosis.
It's a 3 to 4 month transformation, and it's driven by them reaching a specific energy threshold. usually once they hit a minimum length of about 120 millimeters, and it is a dramatic biological event.
The whole body plan changes.
The entire thing. Their digestive system shifts completely, well-developed eyes form, giving them sight. The body silvers up. And most dramatically for our story, the multi-toothed oral disc and that rasping, dagger-like tongue develop.
They're turning from a bottom feeder into a bloodsucker.
And that leads directly to stage 3, the parasitic juvenile phase. This is the phase that defines the lamprey in the Great Lakes.
This is the vampire fish stage.
Exactly. This is the blood-feeding or hematophagus stage. Now, in the native populations, the anadromous ones that swim out to the ocean, this stage lasts about 20 to 30 months in saltwater.
But not the Great Lakes.
Right. And this is crucial. In the landlocked Great Lakes populations, they complete this entire parasitic phase in freshwater. It lasts 12 to 20 months. This difference, this capacity to survive that parasitic stage entirely in low salinity freshwater, that's the foundation of their success as an invader.
We're definitely going to dig into how they manage that. But first, the final stage, the end of the line.
The adult spawning phase. And they only do this once. They are semelparous.
Meaning they reproduce and then die.
It's a massive terminal reproductive investment. Once they mature, they stop feeding entirely, and they migrate intensely up freshwater streams. They use their new sight and, critically, their sense of smell.
The males build the nests.
The males build nests, or reds, often by moving cobbles around with their powerful oral discs. And then the females lay a staggering number of eggs.
Staggering is the right word. We're talking about...
On average, about 70,000 eggs per female.
70,000 eggs.
Yeah.
That high fecundity is the ultimate biological insurance policy, isn't it?
It is. It's a massive evolutionary advantage for an invading species. When you have high mortality from, predation or environmental pressure, that sheer number of eggs helps offset it. It means even if only a tiny fraction of invaders make it and reproduce, the population can still just explode.
Okay, let's pivot to the invasion itself. The initial introduction into Lake Ontario is a little debated, but the catastrophic spread is not.
No, that's pretty clear. There is this older hypothesis, and it's supported by some genetic evidence, like mitochondrial DNA, suggesting they might be native to Lake Ontario, that they survived since the last glacial period.
And there was some unverified report from the 1830s.
An unverified report from 1835, yeah. It's murky. But the dominant, and I think the most convincing narrative, the one that explains the explosive growth and spread, is the invasion by canal hypothesis.
The canals built for trade, not for fish migration.
Precisely. Anadromous sea lamprey were native all along the Atlantic coast of North America. The first connection that allowed them to reach the Great Lakes watershed was the construction of man-made canals.
The Erie Canal being the big one.
The Erie Canal, completed in 1825, linked the Great Lakes to the Hudson River, which already had lamprey. But the more critical events probably involved these little canal feeder streams built in the 1860s, linking the Susquehanna River drainage.
Another major lamprey habitat.
Right, another major one flowing into the Atlantic. And those feeders linked it to lakes like Oneida and Seneca, which drain into Lake Ontario.
So by the late 1800s, they were established in Lake Ontario. But something was holding the line. Something was keeping them out of the upper lakes for decades.
Niagara Falls. That immense waterfall was the natural, impenetrable barrier. No fish could scale it. The sea lamprey's path of destruction was completely halted.
Until the Welland Canal.
So the Welland Canal was significantly improved in the early 1900s. It was built to allow massive commercial shipping to bypass Niagara Falls.
So the shipping industry unwittingly provided the bridge for the vampire fish.
A direct, high-speed route. And once they bypassed Niagara Falls, the spread was almost instantaneous. It was fueled by the sheer volume of suitable virgin habitat.
What was the timeline on that spread?
They were first confirmed in Lake Erie in 1921. From there, it was rapid. Lake Michigan in 1936, Lake Huron in 1937, and finally, they reached the massive deep waters of Lake Superior by 1938.
Wow. So in less than 2 decades, they had access to the entire Great Lakes, a freshwater system unlike anything else on Earth.
And the resulting impact was not just ecological collapse, it was an economic catastrophe, especially for the multi-million-dollar lake trout industry.
The combination of overfishing and this new predator was just too much.
It was a fatal combination. The sea lamprey, which prefers larger hosts, targeted the commercially valuable fish, and lake trout, the critical apex predator, were just decimated. They were extirpated, completely wiped out from Lakes Erie, Michigan, and Ontario.
And brought to the brink in the others.
Critically low abundances in Lake Huron. Lake Superior suffered severe declines, but it managed to hold on to a core population.
The destructive capacity is hard to comprehend. You mentioned earlier a single parasitic juvenile could destroy 17 kilograms of fish.
That's 37 pounds of fish per juvenile. Now, you imagine you have populations that rapidly reach into the hundreds of thousands. The consumption rate is just unsustainable.
And it wasn't just the commercial fishing fleets.
No, it hit everyone. Sport fishing, indigenous fisheries that relied on these stocks for sustenance, for cultural practice. It was a system-wide collapse.
But the ecological nightmare didn't stop with the death of the top predator. The removal of the lake trout created a power vacuum, which led to the infamous alewife effect.
This is a perfect illustration of a trophic cascade, and it shows how one invasive species can just pave the way for another.
The alewife being another invasive forage fish.
Right. With the lake trout and other top predators gone, there was nothing regulating the alewife population. They exploded, and that led to these massive, frequent die-offs.
I remember reading about those die-offs. They caused real chaos.
They were environmentally disastrous. I mean, you have these massive rotting piles of dead fish fouling marinas, beaches. It crushed property values, ruined the tourism infrastructure, and just decimated local economies that depended on a clean, healthy lake. The alewife effect really solidified the sea lamprey's status as a biological enemy.
Control became an absolute necessity, not just for the fish, but for the entire regional economy. We've established the scale of the crisis. So now we have to ask, how? How did this ancient jawless fish, which naturally spends most of its feeding life in the high salinity marine environment of the Atlantic, suddenly thrive in the massive low-on freshwater of the Great Lakes?
To succeed, the sea lamprey had to navigate what scientists call invasibility filters. First, you have the abiotic filters. Could it handle the low freshwater ion content? The temperature swings.
And then the biotic filter.
Exactly, the biotic filters. Was there enough prey, and could it outcompete or survive existing predators? The sources we have strongly support the core hypothesis. They didn't need major genetic or phenotypic changes.
They were already equipped for the job.
They were pre-adapted. Their pre-existing, deep-seated physiological adaptations gave them the capacity to invade successfully from day one.
Okay, let's look at the first key adaptation, just dealing with the freshwater itself. That is a major osmotic challenge. The term you used earlier was facultative anadromy.
Facultative anadromy. It means they have the option to complete their entire life cycle in freshwater, even though their ancestors and many current populations prefer that marine parasitic phase.
And that's a huge physiological feat.
It's immense because, to a fish, low-ion freshwater is constantly drawing the ions, the salt, out of their body. They're constantly losing critical salts like sodium and chloride, and they have to work incredibly hard just to retain water and absorb ions from the environment. It requires high-capacity osmoregulation.
So how does a fish that's used to high salinity water suddenly cope with that constant loss of salt in freshwater? What's the mechanical solution?
The solution is in their specialized gill machinery. Like anadromous fishes, salmon is the classic example. Lamprey gills undergo this preparatory adaptation. They retain these highly specialized cells called intercalated ionocytes.
Sometimes called chloride cells.
Right, chloride cells. And these cells contain dense amounts of an enzyme called Na plus K plus ATPase, or NKA pumps.
Okay, that sounds incredibly technical. Can you simplify what that NKA pump is doing?
Think of the NKA pump as a highly specialized, very busy little vacuum cleaner that's embedded in the gill tissue.
Okay.
In freshwater, its job is to actively suck up tiny amounts of sodium and chloride from the surrounding water and pump them back into the lamprey's blood. It's constantly working to counteract that passive loss.
And the fact that the landlocked Great Lakes population still have this high-powered gill machinery tells us something.
It tells us that the physiological capacity to live and feed entirely in freshwater was already hardwired into their system. It wasn't some new evolution. It was a pre-existing option that they just activated.
And their unique diet, their blood-feeding habit, acts as a sort of secondary buffer against this osmotic stress.
It's a critical piece of the puzzle, and it's often overlooked. Their parasitic diet, which is more than 98% blood, played a huge role in helping them with their ionic and osmotic homeostasis.
Why? What's in the blood?
Well, the blood of their prey, the teleost fishes, is comparable in osmolarity and ion concentration to the lamprey's own internal fluid concentrations.
So you're saying their dinner is essentially their high-end sports drink?
In essence, yes. By ingesting this highly nutrient-dense, ion-balanced fluid, The sea lamprey could effectively counter the continuous ion losses that were happening across their gills. It's an immediate high-volume injection of exactly the salts and fluids they're struggling to retain.
It's a perfect internal chemistry alignment between the predator and the prey.
And that made long-term parasitic survival in freshwater entirely viable. It enabled the invasion.
That's a phenomenal biological hack.
Yeah.
Let's move to adaptation 2, the vampire mechanism itself. We need to clarify their predatory status first.
That's important for you to understand. While they are often called parasites, which sort of implies a reliance on the host staying alive.
Right, a true parasite doesn't want to kill its meal ticket.
A true parasite minimizes harm. The sea lamprey is highly destructive. We already noted that one juvenile can kill 17 kilograms of fish. They rely on multiple hosts throughout their feeding phase, and they rarely return to the same fish. They're more accurately classified as predators.
And they are truly generalist predators.
Completely generalist. In freshwater, they're documented feeding on at least 50 species of fish, everything from large lake trout and salmon to smaller whitefish. And in their native marine environment, they're so effective they can penetrate the tough dermal dentical armor of a lasmo branch.
Sharks and rays.
Sharks and rays. They are built for mass destruction across species lines.
And their secret weapon in achieving this is that complex compound secreted by their buccal gland. Lamphridin.
Lamphridin is the biological cocktail that ensures blood flows freely from the wound and that the host doesn't put up an adequate defense. It is a marvel of evolutionary chemistry, and it's been verified through detailed proteomic and metabolomic studies.
So what's in the cocktail? What are the key components?
Well, let's start with their anti-clotting strategy. They use multiple redundant anticoagulants. For instance, they secrete cathepsin D, which directly breaks down fibrinogen, preventing the formation of fibrin clots.
But they also have a backup.
They also secrete compounds like serpin, which acts higher up in the coagulation cascade.
Okay, give us an analogy. What is that serpin enzyme actually doing inside the fish's wound?
Serpin is essentially a highly targeted wrench thrown into the coagulation assembly line. It specifically inhibits something called factor Zeta, a crucial enzyme needed early in the process that tells the blood to start clotting.
So that are attacking the process at both the beginning and the end.
Exactly. By inhibiting both early factors with serpin and later products with cathepsin D, the lamprey ensures the wound cannot be sealed. They're just maximizing the efficiency of their meal.
But keeping the blood flowing isn't enough. They also have to neutralize the host's reaction.
Precisely. So lamphridin also includes proteolytic enzymes to help with tissue breakdown, but also key compounds with anesthetic and immunosuppressive properties.
So the fish doesn't even know it's being eaten.
It suppresses the host's immune response and prevents the fish from immediately realizing the extent of the damage or, actively trying to dislodge the lamprey. On top of that, they secrete prostaglandins and kynurenines, compounds that are hypothesized to cause smooth muscle relaxation, which just promotes even more blood flow from the wound site.
It's a multifaceted chemical assault.
It is a sophisticated, redundant, and highly generalized predatory mechanism. It meant almost any fish in the Great Lakes was a viable host.
And that capacity was present before they ever entered the lakes.
Which greatly accelerated their destructive capacity once they arrived.
Let's move to adaptation 3, the thermal filter, or the thermal shield. The Great Lakes Basin sees massive temperature variations, from ice cover to summer heat. Did this filter pose any real barrier to a fish that lives from northern Florida to Labrador?
It posed very little barrier. Again, this comes down to their pre-existing physiological resilience. Their native range exposed them to extreme thermal variation, from below 0 Celsius to the mid-20s in the summer.
And they're considered a cold, cool water fish.
They're classified as cold, cool water gillfishes, yeah. The larvae prefer a thermal niche between about 17.8 and 21.8 degrees Celsius. This tolerance just allowed them to slot right into the existing Great Lakes tributary thermal regimes with ease.
But the key scientific concept here is their low thermal plasticity. We need to fully define that, because it's critical to their resilience and to predicting their future under climate change.
Thermoplasticity is the ability of an organism to adjust its critical temperature tolerances based on the environment it's currently living in. Most organisms can adjust their internal thermostat up or down to better suit the conditions they're acclimated to.
And the sea lamprey is terrible at adjusting its thermostat.
Yes. They exhibit remarkably low thermal plasticity. We measure this using something called the acclimation response ratio, or ARR. Think of ARR as the fish's ability to move its internal thermostat.
So an ARR of 1.0 would be a perfect adjustment.
Right. An ARR of 1.0 means if you raise the environmental temperature by 1 degree, the fish can raise its lethal tolerance limit by 1 degree. The sea lamprey's ARR is extremely low, between 0.075 and 0.12.
So if the water warms by 10 degrees, they might only shift their tolerance by 1 degree.
Precisely. Their internal thermostat is practically bolted down, but, and this is the critical takeaway, it was bolted down at a high setting from the start.
So they didn't need to be flexible.
Exactly. Their upper incipient lethal temperature, the point where death begins, is already high, around 31.4 degrees Celsius. Their critical thermal maxima, where they lose motor function, is up around 33 to 34 degrees. They were just born with a high resilient temperature threshold. The temperature filter of the Great Lakes was easily overcome.
Okay, finally, adaptation 4, the pheromonal navigation system. This provided a genius evolutionary shortcut for colonizing new areas. Why is their most suitable river strategy so much better for invasion than, say, a salmon's homing strategy?
Well, a salmon is restricted. It has to find one specific historical stream, its natal stream. If that stream is blocked or polluted, that entire population can die out.
But the lamprey isn't so picky.
The lamprey's approach is non-homing. It's a most suitable river strategy. They are wired to seek any river that offers clean gravel for spawning and an adequate larval habitat. This greatly reduces reproductive bottlenecks and allows them to colonize successfully even if the initial invasion force is small.
So how do they locate these suitable streams?
It's a two-step process. First, they use something called barokinesis.
Which is sensing water pressure.
Right, the ability to sense water pressure changes. They use that to locate the general shoreline area where streams enter the lake. Then, once they're near the shore, they switch to their hypersensitive olfaction to detect chemical odorants, specifically migratory pheromones.
And here's the crucial shortcut that enabled that rapid spread after they got through the Welland Canal. The initial invaders couldn't rely on the pheromones of their own larvae to attract more adults. There weren't any.
Exactly. In the early stages, the number of invading sea lamprey, what we call the propagule pressure, was low. But lampreys, because of their ancient shared ancestry, share a common suite of these migratory pheromones.
So they could smell the native lampreys that were already there.
The invading sea lamprey could utilize the migratory pheromones secreted by native lampreys, like the silver lamprey or the American brook lamprey, which were already widely distributed in the tributaries.
So the native lampreys were inadvertently ringing the dinner bell, signaling, hey, this river is perfect for spawning habitat.
That is precisely the narrative. This use of heterospecific cues from a different species, compensated for the low numbers of invaders. It allowed the early colonizing sea lamprey to identify prospective rivers immediately, granting them a massive head start that enabled rapid colonization and system-wide spread.
Okay, so we have an incredibly resilient pre-adapted invader. Now let's look at the war fought against it, a control program that has been remarkably successful, precisely because it ignores the lamprey's strength and exploits its specific physiological weaknesses.
The Great Lakes Fishery Commission, or GLFC, was established in 1955 to manage this, and their control program has suppressed sea lamprey populations by over 90% from peak levels.
Which has allowed the fish stocks to rebound.
And supported that multi-billion-dollar economy. This success is a textbook case of fighting biology with better biology.
And the backbone of that success is Control Tactic 1, chemical warfare. And it targets the most vulnerable stage, those burrowing larval amycoids.
That's right. Because the larvae are sedentary and they're concentrated in the streams for up to 7 years, they are the ideal target. The primary chemical weapon is TFM, or 3-trifluoromethyl-4-nitrophenol.
And it's selectively toxic.
Selectively toxic to sea lamprey. They also often mix it with a second chemical, niclosamide.
What's the purpose of that additive? Why not just use more TFM?
Cost, mainly. Niclosamide is used as a cost-effective synergist. They add a very small amount, like 0.5 to 2% of the TFM concentration, and it increases the TFM's toxicity. That allows them to use less TFM overall, which cuts costs.
And niclosamide can be used in other ways too, right?
Yeah, it's also used in a time-release granular form. That's for treating large connecting waterways or estuarine areas where a traditional liquid TFM application is less effective or just too expensive.
Now we get to the absolute core of why this control works, the mechanism of selectivity. Why does TFM obliterate the lamprey, but generally spare commercially important fish, like trout or salmon?
This selective toxicity relies on a fundamental metabolic difference that's rooted deep in their evolutionary history. The presumed way TFM kills the lamprey is by disrupting oxidative phosphorylation.
The process cells use to make energy.
Right, the process all cells use to generate high-energy molecules. This leads to rapid energy depletion in key tissues like the brain, the liver, the muscle, and you get this crippling buildup of toxic anaerobic waste, primarily lactate, which causes death.
So the question that obsessed researchers was, why does this poison completely ignore the trout?
And the answer is deep in the lamprey's ancient dinner plate. Fish like trout, salmon, bluegill, they survive because they possess a robust chemical defense mechanism, detoxification. Specifically, they use a phase 2 biotransformation process called glucuronidation.
Glucuronidation. That sounds like a big chemical cleanup operation.
That's a great way to put it. Glucuronidation uses specific enzymes, EDP glucuronidyltransferase, or UGT enzymes, to attach a sugar molecule to the TFM molecule.
And what does attaching a sugar molecule do?
It makes the TFM highly water-soluble, and once it's water-soluble, it can be safely excreted from the body, usually through the bile. The bony fish are just highly efficient at metabolizing and passing the TFM before it can disrupt their internal energy systems.
So the trout can process the poison. The lamprey, however, cannot.
Precisely. And transcriptomic research has mapped this difference clearly. A highly TFM-tolerant fish, like the bluegill, has genes that code for 3 UGT families and 7 different isoforms, including the crucial UGT1 family.
And the lamprey.
The sea lamprey, in contrast, expresses genes for only 1 UGT family, UGT2, with only 4 isoforms. They lack the necessary diversity, especially that UGT1 family, to effectively execute glucuronidation on phenolic compounds like TFM.
That is a staggering biological divergence. We have to circle back to the diet. Why did this ancient fish skip this specific evolutionary defense?
It connects directly back to their lifestyle choice, made over 160 million years ago. Hypercarnivory.
They're meat eaters, or blood eaters.
Exclusively. UGT enzymes are widely believed to have evolved in vertebrates primarily to counter the phytochemical defenses found in plants, to protect against the toxicity of eating herbs. Since the parasitic lamprey is almost exclusively blood and tissue feeding, they face significantly less selective pressure to evolve or retain the diversity of UGT enzymes needed to detoxify plant compounds.
And that metabolic deficiency, born from their specialized blood-only diet, is their modern fatal weakness.
It is. The GLFC is winning this war by exploiting an ancient dietary preference. But applying TFM is not like spraying weed killer. Control agents have to constantly monitor and adjust based on the water chemistry.
Right. Why is the water chemistry so important for TFM effectiveness?
Because TFM toxicity is inversely related to pH and alkalinity. A lower pH increases the amount of the active, free phenol form of TFM. This free phenol is lipid-soluble, which means it easily crosses the lamprey's biological membranes, increasing its potency.
So if the stream's pH shifts, the dose has to shift.
Immediately. It demands constant calibration and adjustment of the dosage to ensure lamprey mortality without harming non-target species. It's an intensive, real-time environmental monitoring job.
Let's move to control tactic 2. Physical barriers and trapping, which exploits a physical weakness rather than a metabolic one.
The lamprey, despite its powerful attachment disc, is a relatively poor jumper and swimmer when you compare it to a salmon or a trout. And this is the weakness we exploit with physical barriers.
They're not building huge dams here.
No, not at all. Most intentionally built barriers are low-head structures. Often, they're designed to maintain only a 45-centimeter drop during the critical spawning run.
So just a small waterfall is enough to stop 70,000 eggs from being laid upstream.
Precisely. The core function of these barriers is to prevent upstream migration. This drastically reduces the length of streams that need expensive TFM treatment, and that's what makes the entire control program economically feasible.
And many of these barriers weren't even built for lamprey control initially.
Hundreds of them were originally built for other purposes, flood control, power generation, and they were just repurposed because they fortuitously serve this control function.
Traps are also integrated into these barriers, but we know removal alone isn't enough to control the population. What purpose does trapping serve today?
Trapping is absolutely critical for assessment.
Not for control, for assessment.
Right. While removing a few thousand adults won't stop the babulation explosion, that massive fecundity means you need near total removal. The traps provide essential data. They quantify adult abundance trends, they estimate damage to fisheries in specific areas, and critically, they help determine where and when chemical control efforts should be targeted next year.
They're the eyes and ears of the control program.
That's a perfect way to put it.
Okay, finally, control tactic 3. Sterile male release, or SMR. This is another highly targeted, species-specific approach.
The concept is classic. You saturate a population with sterilized males who then compete with fertile males for mates, ensuring those females lay clutches of non-viable eggs.
How is this actually implemented?
They used a chemical sterilant called bizazir, and it was notably deployed in the St. Mary's River from 1997 to 2011.
Why the St. Mary's River specifically?
Because it's enormous. At the time, treating the St. Mary's River with TFM was deemed cost-prohibitive and impractical due to its size and flow. SMR offered a viable biological alternative.
But it has some big constraints.
Significant practical constraints. Bizazir is a powerful mutagenic agent.
So, a safety risk for the staff handling it.
A major safety risk. It required a highly specialized, dedicated facility and an auto-injector system to minimize staff exposure. That makes the process geographically and economically constrained.
And it doesn't work everywhere.
No, the technique only works effectively on low-density populations because you need enough sterilized males to overwhelmingly outnumber the fertile ones. It was eventually phased out in the St. Mary's, replaced by targeted granular niclosamide, but SMR remains a viable option for smaller satellite populations that are close to that sterilization facility.
So we've established how resilient the sea lamprey is. Now comes the biggest challenge. That very physiological resilience that allowed the lamprey to invade the Great Lakes is now likely to help it survive and possibly thrive under climate change.
And that could potentially undo decades of control efforts. It's the complex reality facing the GLFC. Climate models are projecting significant temperature increases in the Great Lakes basin. Air temperatures could rise by 1 to 7 degrees Celsius by 2100.
Which means warmer lakes, warmer streams, longer growing seasons.
Longer growing seasons, less ice cover. All of these factors favor this cold, cool, water-tolerant invader.
So how does warmer water accelerate the problem for control agents?
It accelerates their life cycle dramatically. Warmer water leads to faster larval growth, so that larval development stage shrinks from that typical 3 to 7 year duration.
Which means metamorphosis happens earlier.
Earlier and faster. And that requires control agents to apply lampricides more frequently to hit the rapidly developing populations. That means higher operational costs and higher logistical complexity.
And it also produces larger, more destructive adults.
Yes, the extended, warmer growing season results in larger adult sea lampreys. And this isn't a theory, it's already observed data. In Lake Superior, for instance, they recorded it exactly. This could necessitate adding hundreds of kilometers of streams to the already massively expensive TFM program. It could potentially make control economically unsustainable in some regions. And on the other hand, extreme droughts can also undermine TFM effectiveness because low flow complicates the chemical dispersal and monitoring.
And their inherent thermal resilience might actually shift their population centers northward.
That's a key prediction. Because the lamprey's thermal physiology is so robust, a warming environment might favor a northward shift in their optimal habitat. This could lead to greater colonization and population growth in the deeper, higher volume Upper Great Lakes Huron, Michigan, and Superior assuming suitable spawning habitat is available.
Which puts even more pressure on the largest, most ecologically sensitive parts of the basin.
Exactly.
So given these compounding threats, control research is shifting toward emerging, highly targeted solutions, moving away from broad chemicals and toward ultra-specific biological manipulation. Let's start with chemosensory manipulation.
This targets the sea lamprey's most developed sense, olfaction, its sense of smell. Their olfactory system is incredibly acute. A large proportion of their brain structure is dedicated just to interpreting scents.
How sensitive are we talking?
They are acutely sensitive to bile salts, which function as pheromones. They can detect them in the pico to femtomolar range.
Which is an unbelievably tiny concentration.
Almost unbelievably tiny. So if we know they can smell that well, we can exploit that sense.
What progress has been made using specific pheromones?
The most immediate practical success has centered on one component of the male sex pheromone. It's called 3-KPZS.
3-KPZS.
Researchers successfully tested this compound as a synthetic lure to bait and enhance trapping efforts. It resulted in an average 10% increase in trapping efficiency.
A 10% increase might not sound like a huge number to a casual listener, but given the scale of the Great Lakes, that's a significant return.
It's highly significant in an integrated pest management program, and that success was enough for 3-KPZS to be registered as a vertebrate pheromone biopesticide in both the U.S. and Canada. That's a key regulatory milestone. It allows for its widespread sanctioned use in control programs.
But the ultimate goal is the ultimate lure. What's missing from 3 key PZS alone?
Well, we know that the full eau de bouquet produced by a mature male is significantly more attractive than just that one component. So the goal is to identify the entire multi-component migratory and sex pheromone blend to create super lures, lures that could dramatically increase trapping efficiencies.
And maybe even enough to impact recruitment rates directly.
That's the hope. And we are also exploring alarm cues.
Alarm cues, something that tells them to stay away.
Exactly. Researchers are identifying chemical signatures like the odor of putrescine, which is released from dead or dying lampreys, that could be used as a targeted repellent. Imagine using a synthesized putrescine derivative to repel lampreys from key high-value spawning streams. You could effectively steer them away from sensitive areas or into controlled trapping zones.
The final, most futuristic approach is genetic control. This seeks to exploit those deep physiological choke points using gene editing.
This is the frontier. It's made possible by the sequencing of the full mitochondrial and nuclear genomes of the sea lamprey. The initial work focused on gene knockdown.
Using molecules like morpholinos or RNA interference.
Right, RNAi. Using it to inhibit genes that are critical for survival or metamorphosis. The breakthrough here is that RNAi has been successfully used in lab settings to increase larval mortality, and crucially, it was shown it could be delivered via food.
Which is a potentially feasible large-scale delivery mechanism. You're not relying on injection.
Right. And then, of course, there is the gold standard of genetic manipulation, CRISPR.
The CRISPR-Cas system.
It's been adapted for use in lamprey species, allowing for highly precise, targeted gene dilution. The long-term, most impactful strategy being explored is sex ratio distortion technology. This is often called daughterless technology. It's similar to efforts in other invasive fish like common carp.
Explain daughterless technology. How would that work in practice?
The aim is to genetically modify a male so that it only produces male offspring. If you introduce enough of these modified males into the wild population over several generations, you theoretically shift the sex ratio entirely, causing the population to collapse completely due to a lack of reproducing females.
That sounds like a perfect solution. But what are the massive practical and ethical hurdles that prevent us from deploying it tomorrow?
Practically, the challenge is delivery. Many techniques, especially CRISPR, currently rely on injecting the modified genetic material into the developing embryo. That's completely unfeasible for millions of individuals in the wild.
You need a better vehicle.
You need a vehicle like an RNAi delivered via food that can distribute the gene modification broadly. And ethically, the challenge is immense. The risk of the genetic manipulation escaping or jumping to imperiled native populations in the Atlantic or other continents is a major, major concern.
And that brings us right back to the central paradox of this whole deep dive. The Great Lakes work is directly informing global conservation efforts.
That's right. The deep, detailed characterization of the sea lamprey's ecophysiology that North American scientists developed for controlled thermal tolerance, reproductive strategy, chemosensory sensitivity that is now essential for predicting how those imperiled native populations in Europe and the Mediterranean will respond to climate change and habitat degradation. The eradication work is supplying the data needed for conservation elsewhere.
So we have traced the double life of the sea lamprey. It's an ancient, jawless creature whose pre-existing biological toolkit, its resilient thermal tolerance, its high fecundity, the ability for facultative anadromy and survival in fresh water, and that devastating anticoagulating lamphridin made it the perfect, sophisticated candidate for ecological disaster the moment the Welland Canal opened.
And what's so fascinating here is that the Great Lakes Control Program is globally recognized as one of the most successful invasive species programs because it relies almost entirely on that highly specialized physiological knowledge. We are not fighting the lamprey's strength, we are fighting an ancient organism by exploiting its modern metabolic and chemosensory quirks.
The inability to perform glucuronidation.
The poor jumping ability and the hypersensitive nose that can be fooled by synthetic pheromones. Physiology is quite literally the weapon being wielded in this $7 billion war.
The current control is a layered defense lampricides, barriers, pheromones, and soon, potentially genetics, all working ceaselessly to counteract the lamprey's inherent climate-proof biological resilience. And this creates the extraordinary conflict we began with.
The simultaneous existence of an invasive scourge and an endangered species.
In the Atlantic and European streams, sea lamprey populations are threatened due to human factors, habitat degradation, fragmented spawning routes.
And biologists there are now using the detailed ecophysiological data generated in North America, the enemy playbook, if you will, to model population resilience and manage necessary conservation efforts.
So what does this all mean for you? Consider the inherent ethical and biological conflict facing this single globally distributed species. Biologists in North America are working ceaselessly to develop daughterless genetic technologies that could completely crash the population.
While simultaneously, biologists in Europe are trying to characterize that same species' genetics and ecophysiology to ensure its survival against similar environmental pressures.
Oh. And this raises an important question. When facing a globally resilient species with such dramatically different local impacts, can humanity successfully manage a species for eradication in one region while ensuring its conservation in another, without those highly focused efforts inevitably contaminating or undermining each other's goals? That biological balancing act is one of the most pressing environmental questions of the 21st century.